Welcome, everybody. My name is Marcelo Vázquez, and I'm the head of radiobiology section and part of the low dose radiation research at the Canadian Nuclear Laboratories, also known as a CNL. On behalf of CNL and our supporting partners, Atomic Energy of Canada Limited, Health Canada, and the Canadian Nuclear Safety Commission, thank you very much for joining us today. We are so excited to have Dr. Douglas Boreham present today as part of our low dose radiation webinar series. The goal of this webinar series is to help strengthen connection between the research community by engaging world leaders, researchers pertaining to the effect of low dose radiation from different sources and also with a multidisciplinary approach. If you have joined us before, that at the end of Dr. Borham's presentation entitled Biological Effect of Low Dose Radiation from Deep Earth to Deep Space, you will have an opportunity to ask questions. Please use the Q&A function on your screen to write in these questions. And now I have the privilege to introduce today's esteemed speaker. Dr. Douglas Boreham received his PhD from the University of Ottawa in 1990. He was a research scientist at the Atomic Energy of Canada Limited at Chalk River, CNL, for 10 years before he became a professor at McMaster University, where he worked from 2000 to 2012. He's currently a professor at the Northern Ontario School of Medicine University and is the head of their Medical Science Division. While working at McMaster University and Nottingham University, he also held positions at Bruce Power as principal scientist for the Environment and Integration departments. Dr. Boreham is a recognized leader in the areas of low-dose radiation effects in medicine and health and radioprotection of humans and environment. In 2012, he was selected as an expert Canadian delegate for the United Nations Scientific Committee on the Effect of Atomic Radiation, one of the more prestigious committees in the world. He earned several awards over the course of his career, including the McMaster President's Award for Excellence in Instruction in 2004, the Canadian Achievement Award for Outstanding Education and Communication in 2005, the Canadian Radiation Protection Association's Distinguished Achievement Award in recognition of outstanding contributions in the field of radiation protection in 2009, also the Radiation Research Society's Mentor of the Year Award for Scholars in Training in 2010, and in 2015, the International Dose Response Society selected Dr. Boreham as a recipient of the Outstanding Leadership Award in the field of dose response. But it's not over. He also supervised numerous graduate students and postdoctoral fellows and has over 160 peer-reviewed scientific publications. Dr. Boreham is also a board member of the Radiation Research Foundation and the 2024-25 president for the Radiation Research Society. So with this, I would like to introduce our president, Dr. Douglas Boreham, and Doug, it's an honor and a privilege to have you today, and looking forward for your presentation. Thank you.
Thanks, Marcelo. Appreciate that great introduction. It's I really thank you for the invitation to give this seminar, and I'm looking forward to getting some interesting questions at the end. So without further ado, let's start the show. So of course now I'm gonna go forward. Oh, yeah, something go wrong here. I get my little thing to show here. Slide, reduce that. There we go. All right, sorry for that slow start, but I guess I had to put the other presentation on. So again, my title is Radiation Effects from Deep Earth to Deep Space. And I am from the Northern Ontario School of Medicine. You can see down in the left corner, there's a picture of like a mine shaft with some kind of coating on the inside. And that's our sort of deep earth experimental facility I'm going to talk about. And on the right is a little picture of a small spacecraft out in deep space heading past the moon. And I want to talk about that a little bit towards the end with respect to the research we're doing in collaboration with NASA. So I'll start off with this nice or this silver dollar coin that says Elliott Lake, Ontario, Canada, the uranium capital of the world. And so that's where I started my career in radiation science, because I was born and raised in Elliott Lake. And my father was a uranium miner. There were 9 operating uranium mines in Elliott Lake at the time when I was living there. And he was a uranium miner for several decades. And of course, I went to Elliott Lake Secondary School, which is the home of the atoms. So I think you're starting to get a feel for why I've got a little bit of interest in radiation in my career. So I've been a radiation biologist for about 39 years, and I have experience in health and environmental effects. And on the right is a PET scan of a human body where you can see a large tumor in the one lung, the right lung of this individual. And this happens to be my father, who again was a uranium miner, and he did smoke, and he did a lot of other things. He had silicosis because of all the silica dust he was breathing and mining. He was also subject to inhaling aluminum powder, which is called McIntyre powder, which all miners at the time had to do. So his lungs took quite a beating, and eventually he succumbed to getting a tumor in his lungs. And a lot of people ask me, what do I do for research? And I'd rather say, why do I do research, the type of research I do? So it's not what do I do, it's why do I do it? And this is one of the reasons why I do it. I'm from Elliott Lake. I've been raised around the whole idea of uranium and the risks of radiation. And I have a father who's a uranium miner who died of lung cancer. So that's why I do this type of research. I need to find the answers that will satisfy my curiosity. The other reason I do it is because last couple of weeks ago, I got this little thing in the mail saying I should go get my house tested for radon, because radon is, it's a deadly gas. It's undetectable by sight and smell. It's the #1 cause of lung cancer in non-smokers. And so these types of things I tend to think about and actually say, well, I don't know if that's necessarily true. I'm going to test these things. So if you Google several years ago the image for radon gas, this little guy came up, and I put it up to remind me to tell you a story when I was growing up in Elliott Lake, when a surveyor came to our house, measured the radon levels in the basement, and said simply that, oh, well, the radon levels are high like everyone else in Elliott Lake. As long as you don't stay in the basement more than 2 hours a week, you shouldn't have any real concerns with the radon gas, and we'll try and mitigate what's in there. Well, that evening I was sitting at the supper table talking to my parents, saying, you do realize that my bedroom is in the basement and I sleep there every night. So you can imagine what runs through your head when you're 14 years old, saying, okay, so I shouldn't sit, what's happening to me? Do I look like this little man, or I'm going to turn into this little man? So following that up, we have started radon research. We built a radon facility in Flinders University with collaboration in Australia. This is a picture of me with one of the guys that built the instrument. The instrument can house animals that will breathe radon gas and other types of particulates. So we'll understand what the real health effects are. And we're working with collaborators right now. We've done experiments, and I'm not going to talk about that research now, but just to let you know, that's why I do the research. I want to talk to you today about adaptation to stress. And adaptation to stress is a universal response of all our cells. Pretty well every organism, every cell, it responds to stress in different ways, and it adapts itself to subsequently resist further challenges from different stresses. And this is an evolutionary conserved response. Like I said, this is a defense mechanism. I put that little word cloud I made in the shape of a fighter jet, just to say this is a defense mechanism found in all organisms and all living cells that we know of. But before we get started into the deep space and the deep space and the underground research, I want to set the stage a little bit for what I'm talking about when I mean adaptive response. And early on in my career, when I just finished my PhD, I was working with Ron Mitchell, who unfortunately, regrettably, passed away this week on March 17th. But Ron was my PhD supervisor and mentor and colleague throughout my entire career. And early experiments we did with yeast, and here on the left-hand side is a nice picture of grapes here. These are Vidal grapes in my vineyard I had when I worked at McMaster. And on that surface of these grapes are these cells, which are yeast cells, Saccharomyces cerevisiae. And those yeast cells are the cells that we do a lot of research on because they're eukaryotic cells, cells very similar to our cells in terms of DNA, genes, and metabolic functions. This graph from that pit that Ron had published back in 1982 shows that you have survival on the left-hand side, and you have on the bottom dose of radiation, and this is dose in kilogram because yeast are very resistant organisms to radiation. But if you look at the curve here, this bottom curve with the squares, that's survival curve of yeast that not have been stressed out. And the second curve up a little higher, which shows the yeast are becoming very resistant, this curve here. What happened is the yeast are normally growing at 23 degrees Celsius. If you transfer them to a water bath where the temperature is increased to 38 degrees, they start adapting to that temperature shift, going into a heat shock response. And then only after 20 minutes, you can see that survival to those doses of radiation has significantly increased. The top line is the survival of yeast that have been sitting at 38 degrees for 1 hour. A significant 2 orders of magnitude increase in radiation resistance due to heat-induced stress. Well, that can carry on to other organisms, and this is an example of heat-induced radiation resistance in mice. And on the right-hand side is the reference for that. It was done in 1991 by Shen et al. And its whole body hyperthermia is a potent radioprotector in vivo. So what they did is they took a group of 200 mice and they gave them a whole body hyperthermia treatment. In other words, put them in a sauna and basically heated their body up to 40 degrees Celsius for 1 hour, 24 hours prior to being irradiated with a large dose of radiation, a 9-gray dose. So mice normally have a body temperature of 39 degrees. So increasing their body temperature by 1 degree for 1 hour, 24 hours before giving a 9-gray dose of radiation. Now, 9-gray to this strain of mice is what we call an LD 100, so 100% lethal dose to mice after 15 days. So LD 115. So 15 days after these mice get 9-gray of radiation, 100% of them are dead. If, however, they were first given this heat shock 24 hours before, this is what happens. No mice dies. So they become significantly radioresistant. So heat-induced radiation resistance is a real thing in mammalian cells and in organisms. And if you notice the bottom of the timeline, that's 90 days the last time point. So 3 months after getting a potentially lethal dose, these mice are still alive. So what we did at CNL back in 1999, we published a paper where we asked the question, can we adapt mice using hyperthermia to actually prevent cancer formation? And this is an experiment we published for myeloid leukemia in a genetically normal mouse. You can see that this line here represents a 1-gray dose given to mice, and then they're allowed to live out their lifespan. And the line represents the survival of the mice that got myeloid leukemia, where the last mice dies around 600 days. Well, the next line, we actually gave the mice before the 1-gray myeloid leukemia inducing dose, we gave them a 1-degree temperature shift. So we gave them a fever of, again, 1 degree, 40 degrees for 60 minutes, 24 hours prior to the 1-gray dose. And you can see then that the mice, they did get myeloid leukemia, but they gained back half their life expectancy. So if you think about cancer risk, and you think about things that can extend your life expectancy, the ultimate goal in cancer therapy or in preventing cancer is to keep you alive long enough until you die of something else. So if you extend the latency period of cancer out far enough, you won't get cancer, you'll die of something else. So there's another example of heat-induced genomic stability that you can show that actually mice do not get cancer after getting stressed by heat. Well, the next step is when I was working with Ron Mitchell again back in 1991 after my PhD, we looked at radiation and how radiation can induce the same sort of stress response that we see with heat, in turning on mechanisms such as, can we induce radiation resistance using heat? With radiation, can we induce radiation resistance? Can we use radiation to actually prevent mutations from forming? So this graph here on the left shows you that this is the number of hours of incubation that the yeast get after getting exposed to a 100-gray inducing dose. And that sounds large for us, but again, remember, yeast are very resistant to radiation. So 100 gray is given to the yeast cells either in the presence of nitrogen or nitrous oxide. and they're allowed to incubate with time, and then they are challenged with a 1 kilogray test dose. So survival to 1 kilogray is down here without any adaptation from radiation. But if we give 100 gray and wait 1 hour, 2 hours, 3 hours, the resistance to radiation starts building in these yeast, and until they get again up about 2 orders of magnitude resistant. And the difference between the two lines is simply that nitrous oxide, when you radiate in the presence of nitrous oxide, you get twice the yield of for hydroxyl radicals. And so it looks like hydroxyl radicals are one of the mechanisms to signal this particular process. The same thing happens with mutation. If we treat the yeast with a carcinogen, an alkylating agent called MNNG, it creates this number of mutations in mice per 10 to the 5 cells. But again, if we give them doses of radiation and allow them to incubate for 30 minutes, the mutation frequency goes down significantly out to that response. So this dose of radiation, this larger dose of radiation right here at 40 gray and allowed to incubate for 30 minutes causes a significant reduction in mutation. So again, radiation induces radiation resistance. Radiation induces mechanisms that can help repair lesions that are mutagenic in yeast. All right, so what about in cancer? Can we use radiation to extend latency period? This graph should look very familiar to you because it's almost identical to the graph that I showed you earlier, where a 1 degree temperature shift caused cancer latency to go out and actually give back mice half their life. Well, this in fact is a very similar experiment, except this time we gave the 1 gray myeloid leukemia inducing dose. This is simply a 100 milligray, 24 hours prior to the 1 gray dose, and the same effect is seen where you extend the latency period of the cancer and the mice gain back half their life. So heat and radiation, the right type of stresses, can induce these mechanisms that give you the same outcome when you look at cancer risk. Well, some really elegant work done at CNL with Ron Mitchell was done by Ed Azam and Sonia de Toledo and Pete Rapphorst, and it's looking at transformation in vitro. And this is the 10T1/2 cell assay that Ed was an expert at. And simply it's you seed the flask with a number of these 10T1/2 cells, and some of the cells will transform into what we call type 3 foci. And these type 3 foci look like this. They're kind of like a mini tumor growing in the flask. They pile up on top of each other. They start invading the flask, very characteristic of cancer. And if you take these cells and inject them into a mouse, the mouse will develop cancer. So these type 3 foci are very important for understanding transformation in vitro. So Ed did some very nice experiments, lots of experiments in this. And this is one of the results that really impresses me, and that is, if you just look at the spontaneous frequency of transformation in the cell line for every 10,000 cells, with this particular cell line, you're getting just under 20 of these transformers per flask. But if you were to expose, when Ed exposed these flasks to a 1 milligray, a 10 milligray, or 100 milligray dose of radiation, a single exposure allowed the flasks to transform. The spontaneous frequency of transformation dropped significantly. And you can see it's way down there to maybe even 5, going from 20 from spontaneous control levels down to 5. So a big reduction in transformation and genomic instability. The other interesting thing about this is 1 milligray seems to be the same effectiveness as 10 or as 100 milligray. And I would call those 3 doses low dose radiation. The other interesting thing is the 1 milligray dose equates to roughly 1 photon track per cell. And that simply represents, obviously, the lowest dose you can get at the lowest dose rate. You can't get a dose lower than 1 photon per cell. And that's the lowest dose rate because you're not getting 2. So that's saying that even on average, 1 photon per cell, you can get this effect of transformation suppression. Some experiments, again, done by Ron Mitchell and others at CNL, looked at the same thing in animals developing cancer in a cancer-prone mouse model system, which we call P53. These are P53 heterozygous mice, so they develop spontaneously a number of tumors. And what Ron did was he gave, again, simply like Ed did, a 10 milligray or 100 milligray dose and looked at lifetime survival from tumors in this cancer-prone model. And you can see, without any radiation doses, this is the number of tumors mice get with time. And if nothing's been done, but the green and the red lines represent the 10 and the 100 milligray single dose to these mice, which again gave them back half their life compared to the controls over here. Again, it's supporting the idea that these low doses can stimulate mechanisms that protect us against transformation. So then the question comes, low dose diagnostic x-rays, are we giving ourselves cancer? Of course, my answer is going to be, we think not. And based on a number of things, if you think about CT scans, you're roughly getting about 10 to 40 milligray, depending upon the scan, what you're looking at. This is a classic CT scan my colleague Ian Days gave me at the Juravinski Cancer Center in Hamilton. This is showing a tumor in this individual's lung, showing dark spots for emphysema. And then Ian always points out that this is likely the cause. This is a pack of cigarettes in the individual's chest pocket when he got his scan done. So you kind of got the cause and effect on this picture when it comes to radiation and CT scans. But again, the dose from CT is about 10 to 40 milligray. So a paper came out back in about 2007 in the New England Journal of Medicine by Brenner and Hall that basically concluded that 2% of all cancers in North America over the next decade will be caused by CT scans. So my colleagues, Ron and I, and several others put out a paper in the Journal of American Physicians and Surgeons that said CT scans may reduce cancer risk, and this cancer risk in North America may be reduced by 2% over the coming decades because of low-dose medical CT scans. And so then that starts the debate, and that starts the story of how do we show this is going to work? Well, it starts off with reactive oxygen species and endogenous damage caused to our DNA, and the link is simple. When we breathe oxygen or exercise, our mitochondria produce these things called reactive oxygen species, hydrogen peroxide, superoxide, and hydroxyl radical. When you get irradiated, our cells in our body are mostly water. The water gets hydrolyzed, producing exactly the same type of free radicals, so hydroxyl radical peroxide and superoxide, and that damages our DNA. So we have endogenous effects and we have radiation effects. And what is the balance between what are the natural levels of the endogenous things versus what radiation does? Well, it simply works out that a 10 milligray CT scan would equate to an average of about 10 photon tracks per cell. For every hour, mammalian cells have 50 to 100 times as much spontaneous damage from oxidative metabolism as they would from a 10 milligray dose. So 100 times more spontaneous damage than a CT scan every hour. So there's about 10,000 measurable DNA alterations per hour in each mammalian cell and 100 or fewer from a CT scan. So when people ask me, is this CT scan going to cause a lot of damage to my cells and cause me to get cancer? I try and explain to people that the hour you were waiting before your CT scan, there were 1,000 times more damage to your DNA from oxidative metabolism. That's repaired, or the cells are replaced. And so the answer to that is not likely, not at all. So we were fortunate enough in Canada here to get a grant from the US DOE to do a large animal study to look at what is the lifetime risk in cancer-prone mice of getting a CT scan and a PET scan or combinations thereof. And the groups that we were testing are here on the left. You can see the different treatment groups we had, a very large experiment, thousands of mice. And this is just a picture of a CT scan. This is the skeleton of the mouse. And the same mouse is getting a PET scan at the same time with a PET isotope that shows he's metabolizing a lot of fluorodeoxyglucose. His heart's pounding like heck. He's got a heat pad on his back that's actually keeping warm. And this is a live mouse that's basically been scanned both ways. So we can get an idea of the dose from these things and look at lifetime cancer risk. So the idea is this: CT scans and x-rays are the anti-promotion or anti-progression of low-dose radiation. So we know if we give a larger dose of radiation to these mice here, with some progression, some promotion, we get cancer forming sometime later. If we give a single low dose of radiation, can we actually extend the lifetime of it and extend cancer risk? Well, here's the result of one of those groups. And this is a simple experiment where one group of mice, they got a 1 milligray CT scan at 7 to 8 weeks of age, and they were allowed to live their life, compared to control mice that never got a CT scan. And the overall answer to this is, what happened? Well, a single CT scan did not increase cancer risk in these mice. At 50% survival, they gained about 18 days of longer life, and at 25% survival, about 34 days of longer life. And people say, well, that's not that long. Well, remember, mice have higher metabolism rates and shorter lifespans. And there's research done to equate sort of mouse days to human days. And 9 mice days equal about 1 human year. So you can see at 50% survival, you've got about 2 years of extended life. And at 25% survival, you've got about almost 4 years of equivalent human life sparing by getting a CT scan, a simple 10 milligray CT scan. All right. Well, what happens if you give a large dose of radiation? You give mice a high dose 4 gray and give them cancer, and then ask the question, if we give CT scans after we've initiated cancer, can we also extend latency period and increase lifespan? So the experiment was like this. We gave the large dose, 4 gray dose to the mice. We waited 1 month, which was a lot of debate with me and my grad student about how long we wait after before we start weekly CT scans, 10 of them. So for 10 weeks, we get a weekly CT scan. And so we did that experiment. And again, this is the result. Multiple CT scans did not increase the risk of mice that are getting cancer from a large 4 gray dose. In fact, it did exactly the same thing as a single CT scan. So CT scans after inducing cancer in these mice extended the latency period of the disease, and the mice lived longer, almost to the extent where we got the equivalent of 4 human years worth of life brought back by giving these CT scans. So the bottom line is we didn't increase risk, because if you think about some of the models out there where if you increase risk, if you give 4 gray plus more radiation, you should get more risk, but in fact, we're getting less risk. Here are the two publications dealing with the single CT scan versus the multiple CT scans afterwards. And there's a whole journal article focused on this type of research. And part of the research I'm going to talk about later, the SNOLAB stuff, this radiation research journal contains all that information and all those papers. All right, so now let's get into some of the stuff that we're going to talk about now with respect to the deep space and the underground research. So if you're into models and stuff like that, there's all sorts of models we can try and predict what might happen at low doses where we're not getting large dose effects like we have here, where the A-bomb survivors represent these black dots, and you can draw a straight line to zero, or you can draw the green line, which I tend to do when I do experiments, and I'll show you some more stuff. But the question is, what happens here when you get to below background radiation? If you believe that low doses of radiation are stimulatory, then what happens if you take away background radiation? Is it going to cause negative effects, or is it going to actually be protective? Because if you believe all radiation is bad, then it should be positive. So our working hypothesis is natural background radiation is essential to life, and the absence of background radiation is detrimental to biological systems. Okay, so here's a picture I took in Sudbury back a few years ago over the lake in front of my house. And those, of course, are the northern lights, which are cosmic radiation from space, dissipating the energy in the atmosphere as they come through and interact with nitrogen and oxygen in the atmosphere and giving these nice colors. But nonetheless, the radiation's there, whether you can see it or not. It's always coming towards us, always hitting us on this planet. Some of the types of radiation are going right through our bodies, right through the earth and coming out the other side, and others are shielded by the atmosphere or some layers of material in between us. And in fact, we have a lab that is located underground, and you can see on the left-hand side, there's a little cartoon that basically shows a little lab on the surface with the cosmic rays here, and 2 kilometers down, there's about 50 million times less cosmic radiation down there because of the shielding of norite rock on top. And that lab is called the Sudbury Neutrino Observatory, or SNOLAB. And a guy named Art McDonald, who was working at Queen's University and at CNL, did some work and won the Nobel Prize looking at how neutrinos interact in this detector that you can see the picture on the left, where there's some people standing below it. Right here, the people. So it's a large detector with photomultiplier tubes filled with heavy water. And here's Dr. McDonald at his workstation in the SNOLAB wearing his sort of mining outfit underneath the SNOLAB, and he basically worked out that there was 3 types of flavors of neutrinos, which meant they had mass and they oscillate, and he won the Nobel Prize for that. So the SNOLAB itself, if you just look at it, it's located in Sudbury. It's probably about 30 kilometers from our university. The SNOLAB is located 2 kilometers underground. It's shielded by the equivalent of about 6 kilometers of water shielding, or there's like 50 million times less cosmic radiation, 30 times less natural background radiation. It's a class 2000 lab. And just a cartoon on the right here shows at the surface, you have these mine shafts going down. All these little black lines represent drifts. The yellow represents nickel in the mines. But if you go right down, and this is an active mine, it started as an open pit mine, and then they went to shafts and started going down to get this ore. If you go right down to the bottom, the 6800 level, and you walk down this drift, it takes you to SNOLAB. Well, to get to SNOLAB, like I said, 30 kilometers away. So our students were fortunate enough to get this nice electric car donated by Bruce Power to work at SNOLAB, and we get a nice charging station for free from Bruce Power. And they take this car and they drive out to the SNOLAB, the mine site, and this is the SNOLAB facility where they get all their PPE on and get ready to walk over to this mine shaft where they'll go down with miners and walk down a drift for 2 kilometers, again, to almost 2 kilometers to get to the SNOLAB itself. When you get to the SNOLAB, and this is a picture of me and Chris Tomei and Jake Perkinen, the two guys who basically built this facility initially and got it up and running. And they came up with an acronym because you have to have acronyms when you deal with physicists. In the SNOLAB, it's all mostly neutron scattering or neutron detection, neutrino detection, dark matter detection, things like that. They wanted an acronym, so I believe Jake and Chris came up with this REPAIR acronym, researching the effects of the presence and absence of ionizing radiation or repair. And this is just us with our all full mining gear on, getting to the end of the drift where we have to clean ourselves off, get all this dirty clothes stuff off, get the dust off us, and then head into this Class 2000 clean lab. You can see it's an intricate network of things in SNOLAB. Our lab is located right here where this little circle is, and based on all this intricate networking. And of course, one of our students, I believe it was maybe a combination, but I think our postdoc now, Mike Lapointe, developed this crest. So we have our research in the effects and the presence of absence of radiation. We have a little yeast cell walking down a mine drift, and on the top we have cosmic radiation hitting the surface, and a yeast cell heading down this track we head to the lab to do research on the effects of eliminating cosmic radiation and other background radiation. So I asked Mike to send me a picture of our lab, a new one, because he had up other pictures that were sort of outdated, and he sent me this picture. It's a pretty impressive lab, but that isn't our lab, obviously. This is an AI-generated picture of a lab based on emails and correspondence and papers that Mike have published. This is what the AI thinks that his lab should look like based on what he's been doing. Actually, what his lab looks like is this. Our lab looks like that. A typical lab, it has your standard tissue culture stuff. It's got cell counters and water baths and the standard stuff you need to grow cells and study cells. And I just put the picture of Dr. Revo up there because we always say it's a secret underground lab. And there are sharks with lasers on their head down there, but they're laser pointers, not real sharks. The critical piece of equipment we have down there is called a specialized tissue culture incubator. And this piece of equipment costs a significant amount of time and money to make, but thanks to the SNOLAB people, this is the type of work they do. So they built this incubator that will take cells and other things in there that has all sorts of special attributes that allow us to eliminate things like radon gas, external radiation from the walls, and other types of radiation that might be present in the terrestrial environment. And inside here's a lead casket where our cells will grow, and it's temperature regulated, CO2 regulated. And it has this gas here, which is the air that we put in, which is aged at least a month. So this old air that no longer contains radon in it, and then it's filtered. So we have this ability now to grow things in virtually the absence of all radiation. This is just a timeline that was published a couple of years ago, what we accomplished. We started in 2016 with simple experiments with whitefish embryos, and just to see if they would grow and see if we could get students that would be diligent enough to actually keep going down there and not quit after 2 or 3 weeks, because it takes a long time to get down there and you have a short period of time to do your work and then you got to leave. And we've done a number of experiments from using yeast to human cultured cells to C. elegans. And now we're working down there with the desiccated yeast model system, which I'll talk about. So the first paper that I want to talk about is, again, from Mike was the primary author on it, and Chris is senior author, and all the other people who work down there. Again, it has to do with yeast. It's protracted exposures to what we call anhydrobiotic desiccated yeast. And anhydrobiotic is the term used to say life without water. And so these yeast are desiccated to the point where they're still alive, but they have no water. And there's what they look like before they get desiccated. We don't have pictures of them desiccated, but we're working on that. And simply, everyone has them in their fridge, but these are just freeze-dried yeast that don't need air, they don't need water, they don't need to be fed, and they're alive. So one of the first experiments that Mike did was he looked at what happens if you put these desiccated yeast underground shielded from all this radiation, and two types, a wild type yeast, a normal yeast, and a yeast that's defective in DNA repair, double strand break repair, called RAD51. That's the graph on the right. Well, if you just look at with time, and he's gone out here up to 48 weeks, so it's a lot of work, a lot of going underground, picking up samples and looking at how they survive when you rehydrate them. There's a significant decrease in survival in the cells, whether they're wild type or RAD51, if they're underground, shielded from radiation, shielded from natural background radiation. Now, remember, these are desiccated anhydrobiotic yeast. They're not metabolizing, they're not growing, they're accumulating damage, presumably at a less rate underground where there is no background radiation than they are on the surface. And that phenomenon, or that process, is causing them to be less viable when they're rehydrated on the surface, which is a very striking and interesting result. Well, if you think about the assays that we use, one of the assays we use to look at how the yeast are alive is a metabolic assay, and this is important to remember for next time. I'm just going to check my time here. This is important to remember for when we start talking about yeast in space. So what happens here is on the left-hand side, you have wells that contain yeast cells, probably 1 million cells per well, and they have a fluid in them that basically it reacts to metabolic metabolism and changes color from blue to pink. And if you look at time zero, of course, all the wells are blue because there's been no metabolism. There's no cells in the bottom line here as a control. And then you have different doses of radiation to the cells in triplicate. And if you wait 19 hours, you can see now we're starting to get metabolism in the cells that got zero dose. Of course, none in the ones that have no cells. And you can see as you get increasing dose, you get less and less metabolism, i.e. more cells are dying or are dead until you get 30 grade where there's none. And then if you wait 24 hours, you can see again a dose response based on a colorimetric assay using LMR blue. So using that assay, you can look at things like metabolism in these yeast. And again, this is an experiment where we looked at the ability of yeast to metabolize after they've been underground for these long periods of time, whether it's a wild type yeast or RAD51. And there was no significant difference in this experiment when we looked at the wild type yeast. But again, the RAD51, in the absence of sub-background or in the absence of natural background radiation, the reduction time took longer, which meant the yeast weren't metabolizing as well when they were shielded from the natural background radiation. Well, given that we pretty well eliminated everything possible, there's one other thing that we could eliminate, and that is the food that the yeast eat, or the food that we give to the food that we eat contains natural radioisotopes. And those natural radioisotopes are potassium-40, carbon-14, and a few other ones. But the big one we have is potassium-40 in our body. And human bodies roughly have about 4,000 becquerels worth of potassium-40 decays, which is about 4,000 decays a second in your body every second from potassium-40. So what we did was we decided, well, can we take away the potassium-40 in the media, so eliminate that source of the last sort of source of radiation we can think of. And Mike managed to get some potassium-39, put it in defined media, which had no other potassium. So overall, if you look at doing that particular experiment, this is the amount of radiation they're getting here, experimental nanograys per hour, which means about 835-fold reduction in dose rate by getting rid of everything, including the potassium-40. And in those experiments, we looked at metabolic activity in the same thing, underground in the absence of natural background radiation, and taking away the potassium and the intrinsic potassium-40, we can see that we had a significant effect in both wild type and RAD51, showing that the doubling time of the yeast was significantly increased in the absence of potassium-40 and the absence of natural background radiation, which simply says the more we take away, the worse the cells do when we try and revive them. Same thing happened with growth rate. Again, it didn't happen with the growth rate with the doubling time for the wild type cells, but it did for the RAD51 that were deficient in potassium-40. And so now I'm going to switch gears to a different model system, which actually is relevant to transformation or cancer transformation. And it's the role of natural background radiation in maintaining genomic stability in this CGL1 cell line that we use, which is a human hybrid model system for studying transformation. Very similar to the model system that Ed Azam used to show transformation. This is, again, the cells that turn into they're neoplastically transformed, produce a marker on their surface called alkaline phosphatase, which then allows them to be stained a different color and identify the ones that have become transformed or become cancers. And there's one there on this flask that we can see. Here's what they look like under a microscope, and we count numerous numbers of these foci that are forming and get a real measure of neoplastic transformation under various conditions. The one experiment that was very interesting early on was done by Redpath, Medonka, and others that showed that very similar, in fact, identical to what Ed Azam showed in the 10T1/2 system is if you start with a high spontaneous rate, so it's 0 dose of radiation, here's the transformation frequency per 10 to the 6 cells or 5 cells, and you get about 3. But if you give low doses of radiation, it reduces again. It goes down and down and down until the dose starts getting higher. And then, of course, as higher doses come, you start getting higher transformation. This is your U-shaped curve showing that low doses of radiation reduce transformation in the cell line below the spontaneous rates caused by whatever happens under natural conditions. Well, this is sort of natural background radiation level, 0 natural background. And so our question is, well, what happens to transformation frequency if we actually look at the conditions we can create down in SNOLAB? Well, if we looked at how these cells survived radiation after spending 4 weeks, 8 weeks, 12 weeks, or 16 weeks shielded from natural background radiation, there was no difference in survival. However, if we looked at transformation in these cells, so this alkaline phosphatase activity, which is a measure on the surface marker for transformation, we can see that after 4 weeks, there's no difference between the underground controls and the sub-background controls. If we look at 8 weeks, we can see the difference starting, but at 12 weeks and 16 weeks, there's a significant difference. So the sub-background cells, more are transforming than the ones that are just the underground control. So they're getting exposed to the background radiations down there without the all the other radiation. But the very interesting thing that Jake did in this experiment was very cool. He took the cells that had been down there, so these ones after 16 weeks with a significant difference where you have more transformation in the absence of natural background radiation, put them back on the surface for 2 weeks or 4 weeks, they drop back down. So the surface radiation has actually caused the transformation frequency to spontaneously go back down to normal. So quite interesting result in terms of cancer risk. There's a very neat video about some of this work, and this lab is on YouTube. I put this in here because this is being recorded, this particular presentation, so you'll have this reference so you can actually go watch this very interesting, very fun show of our SNOLAB and our research down there. So earlier on, I told you about finding a living model system to be the best sort of astronaut ever for deep space travel. And that would be an astronaut that doesn't need to breathe, doesn't need any water, doesn't need any food, and does not need heat. In other words, it can withstand the cold of deep space, or minus 270 degrees Celsius. And so what is that? Is there anything that can do that? Well, there is, in fact. Again, NASA is sending some lucky yeast into radiation-filled deep space. And the desiccated yeast model system, or the active dry yeast, is exactly that model system. They don't need air, they don't need water, they don't need food, and we already know that they can withstand minus 196, and we're looking at a way to drop their temperature down to minus 269, which is liquid helium. All right, so on August 29th, 2022, this is our research group. We're down at the Kennedy Space. We're at Cocoa Beach, I believe. We've got a couple of cans of beers with us there. It's about 6 o'clock in the morning, and in the background, we're waiting for the Artemis 1 rocket to take off at 8 o'clock. And it didn't happen. It's actually got scrubbed 10 minutes before it took off. But the beer we had was made with the yeast that we'd grown in the snow, and it was called Cosmic Ray beer because we eliminated cosmic rays, and it was made by 46 North, and the beer was sold, and money was raised for cancer research from the sales of that beer, because we are technically looking at cancer research in the absence of radiation. Well, here we are down there waiting for the Artemis rocket to take off. In the bottom left-hand corner here, you can see me standing by a model of the rocket ship that actually was taken up on the Artemis 1 rocket that contained the desiccated yeast cells that were going to travel into deep space. And the reason I'm wearing this sort of really American type hat was because I got it before a lot of the stuff's going on with the United States and things. But this is the hat that has the STS-118 logo on it from the mission that the Canadian astronaut Dave Williams, who's down in the lower right-hand corner here, right here, was on that mission. So I got to watch the launch basically from the same spot where the launch was right here. And believe me, when they go off, it seems like you're closer than you really are when you can feel the heat coming from them. So the Artemis 1 rocket takes our E cells that we're collaborating with NASA in Ames and takes it into space. And again, here's a bigger picture of the size of it. These are solar panels. This is the actual they call it CubeSat rocket or little spaceship that went up on the Artemis 1. By the way, Artemis 1 finally went up on November 15th when it was supposed to go up on June 22nd when we went down. So here's what's on that little rocket ship. These are microfluidic chip, and that has those little yeast cells in there that are rehydrated at different times while they're traveling into deep space. And you get the colorimetric changes like I showed you before, except it's read robotically and recorded and sent back to NASA so they know how many cells are still surviving as they travel deeper and deeper into space. And of course, our interest in this is on the surfaces. What can we learn from these desiccated yeast in terms of how they function, how they survive, how they deal with cosmic radiation at different levels and different intensities. So I think we checked in November where they were. This rocket, this CubeSat was traveling into space right now as we speak, heading into deeper space. In December, it was about 53 million kilometers away from Earth. I think a couple of weeks ago I looked and Venus was about that far away from Earth at the same time last week. So it's traveling out there. Unfortunately, NASA lost control or lost contact with the robotic fluidic system that would allow us to tell how many yeast are alive. So that data is kind of lost, but it's still recording a lot of dosimetry data as it goes into deep space. So one of the experiments we're doing right now is looking at how these yeast can withstand high levels of neutrons and high doses of neutrons at different rates. And I just put up the NRU reactor and the McMaster reactor because the McMaster reactor basically is where Bertrand Brockhaus won the Nobel Prize, but he also worked at CNL at this time too. And he actually hired the guy on the right, which is Gerald Dawling, to come from Cambridge University and work with him doing neutron scattering. And it was Gerald Dawling that actually, who worked at CNL, wrote the Nobel Prize a nomination for Bertrand Brockhaus to do this work in both those excellent research facilities. And I'd like to say that Gerald Dahl was my father-in-law, or he still is, but he's passed away. So if we look at what happens when we take our anhydrobiotic yeast and put them in the core of a nuclear reactor that has 5 times 10 to the 12 neutrons per second or per centimeter per second, and we look at how long they survive, you can see the survival curve here on this graph, and you can simply see that even up to about 100, sorry, gotta go back, up to about 100 seconds, there's still survival at that flux of neutrons. So we know we're going to learn a lot using this system to understand how these cells deal with neutrons, which are what we're going to have to deal with in space when we're traveling with astronauts and things. So they can withstand a lot of neutron dose. And if you look at sort of comparing this with the photon dose equivalent, so grays and photons, and look at exposure time, you can see that we're getting about 54 gray per second, which is a high dose rate we call flash dose rate, and we're getting doses that are somewhere in the range of 4,000 to 5,000 gray equivalents of neutrons. So I mentioned flash. Flash is a very up-and-coming sort of potential treatment for certain types of cancers because the observation is that a flash radiation, so a super high dose rate, tends to spare normal tissue and kill tumor tissue, differentially kill tumor tissue. So by killing tumor tissue and sparing normal tissue, it gets better treatment outcome. And we did proton irradiations at Triumph with UBC, looking at how Rad51 yeast, these desiccated anhydrous yeast, can withstand flash radiation versus conventional radiation. And you can see, even in this model system, that the same doses, flash radiation is actually sparing the cells more than the conventional dose rates. So we got a lot to learn about on that system too for that. The interesting observation that Mike has made is at the same level of survival, so 6,000 gray, at 55 gray per second in flash radiation versus 6,000 gray at 5 gray a minute conventional, you get the same level of survival, but you notice the growth rate in flash is significantly higher. So the small, tiny colonies on the ones that were conventionally irradiated are suggesting that growth rate has been hindered or damaged by this conventional dose rate experiment, but flash radiation seems to be sparing that mechanism. So that's very, very interesting stuff that we're going to be working on with these desiccated yeast and helping understand how they respond to these doses and dose rates while they're in space. So why do I do this research? Not what do I do? Well, why do I do it? Because all this stuff gets down to the clinic. And I just put this reference up here because we published a paper in the last couple of years where we used half-body low-dose radiation exposures in patients that had recurrent prostate cancer with the idea that we would be stimulating their immune system and would help put their recurrent prostate cancer back into remission. So stimulating the immune systems by giving these patients 150 milligrade twice a week for 5 weeks. And all the data is in this paper. And when you read the paper, if you do, you'll find out that, in fact, this will work in certain patients not on androgen deprivation therapy, but some of the patients are still in remission due to this, using this low-dose radiation to stimulate processes that help slow down cancer growth in vivo. So again, why do I do radiation biology? Because everyone's entitled to their own opinion, but not their own facts. And when you're doing radiation biology, you got a lot of people who have different ideas and different facts and different opinions. I can tell you, I did a lot of work in Australia, and I helped get one of the first uranium mines licensed called Wiluna Mine, which is right here in West. This is a picture of West Australia. Here's a Wiluna mine site. It's a uranium mine. And there's a lot of controversy, and people, especially in Australia, are very radiophobic. And so I would try to go and help educate the regulators, the politicians, and the public on what the real effects are of uranium mining. And of course, I get some delightful emails from people, and this is one I'd like to put up. I think it's horrible that you're in a position of power in your position at a university while peddling lies regarding the health benefits of low-level radiation. If anyone's hurt during the Wiluna mining process, their blood will be on your hands. You're a disgrace to the scientific community, and personally, you should hang your head in shame and remove yourself from your teaching position. So I'm going to tell anyone out there who's doing radiation research, whether students or scientists, that you need a pretty tough skin, because a lot of people have this opinion of radiation as more of a spiritual opinion rather than a scientific opinion. And she signs a letter like, seriously, you're a dickhead, Elisa Garrard. So I want to thank everyone for listening, and I appreciate the fact that you tuned in for this little podcast. This is a picture of us, Ron Mitchell and my wife Jo and I in San Juan, Puerto Rico at a radiation research meeting, so which means I get to give a plug as president of the society this year. For everyone that's listening, to please try and get to Puerto Rico for September 21st to 24th. The registration is open now, and so are the abstract submissions. And again, thanks very much.
Douglas, can you hear me?
Yep.
Well, thank you very, very much for a fantastic presentation. And there are several questions already in the Q&A area. So again, first, let me thank you for a wonderful presentation. And also, I want to say that to the audience that I have the opportunity to visit SNOLAB a few weeks ago. It's an incredible experience. It's a one-in-a-lifetime opportunity to visit some extraordinary construction in the laboratory, and please search about SNOLAB and learn about that, because it's an incredible place to do research and be there. So we have several questions, Douglas. So there is a question regarding pressure effect. So if you have some underground experiments 2 kilometers down versus on the surface experiment, there are some difference in pressure that can confound the effect of radiation or no radiation, or the pressure in both environments are the same?
Yeah, it's a great question. In fact, that's one of the things we not struggle with, but we're cognizant of, and we really want to manage it. And we're actually just talked about some experiments yesterday where we're going to try and see if the different pressures make a difference in the biological responses, at least for some of the endpoints we're looking at. I believe it's about 2 atmospheres or equivalent about 30 foot depth in water. And we're noticing that some of the growth rates that we have underground are slower than on the surface. And one thing might be, well, maybe it's just pressure. So that, in fact, is a great question. And we are doing those experiments, and we're going to be starting them very shortly on where we're going to grow things on the surface with the same pressure that it would have underground to see if it's a pressure differential thing. And that's one thing we got to work on right now. It's a good question.
Thank you. Thank you. More. I will read this question. Considering the divergence in interspecies radiation-induced gene expression, not only between mice and humans, but also between non-human primates and humans, how do you translate the results obtained for low-dose exposure in protecting mice from cancer to humans?
Yeah, this is always our challenge, right? We need to find model systems that are going to try and represent what we have. And, mice are 99% the same as us genetically, even though we don't look like them. They have different metabolic rates. I think what we have to do is we have some research now in humans where we can see that these low doses can adapt human cells and human people. We did some experiments on humans looking at how this whole notion of how we can use heat to change different things in humans, and we showed that the same thing we can see in mice when it comes to, say, treating benign tumors, or in humans with, say, a benign type of skin problem like psoriasis, can be shown in humans and in mice. So we treated humans with hyperthermia treatments and showed the same effects that are happening in mice. So I think as we go forward, we're going to have to get more and more once we get more and more into the sort of omics part of what we're doing, which now we have these powerful tools. I mean, we just heard a talk this week from an individual we invited up that it's getting so complicated that now we can go back and look at the phenomenon we were seeing in mice and looking at the phenomenon we're seeing in humans and use these powerful tools to look at what pathways are turning genes on, what proteins are being made, what type of metabolism is going on, and have AI help us with figuring out the complicated processes that we're dealing with here. But it's always a challenge to take a model system and try and extrapolate to what it might mean in humans. I guess what I can say is at least we know that giving CT scans, either single ones or multiple ones, doesn't increase cancer risk. And in the model system, we're not getting any evidence that says this is bad. So at these low doses. So I think we got to keep working towards that goal. And I agree. I've been doing this for 39 years, and it's always a challenge. How are we going to extrapolate this?
Yeah, there's a lot of fun too, doing this because it never ends the questions. So more questions. This is very interesting. The NCRP has endorsed the findings that 0.1 Siever has been demonstrated in children for radiotherapy to induce around 0.5% cancer induction probability increase. Can you discuss this?
I would say there's probably 100 other recommendations that suggest that these cancer risks are increased. Generally, the epidemiologists, they have to work on models, and depending upon the model they use, it's my understanding you can basically show that there is risk or there's not risk. So I am not an expert in statistics or epidemiology. I'm just an expert in the biology that happens at these low doses. And I think if you look at some of these recommendations and where the data comes from, if you have the knowledge, you look in it, you can say, well, I'm not sure that's exactly supporting what the scientific data is saying. And there's lots of stuff, like if you look at UNSCEAR and other places that would argue the same thing, that we can't start extrapolating risk based on mathematical modeling. It has to be real science.
I agree with that. There is another interesting question. Does the heat promote DNA repair, or is heat preventing the damage from occurring? Is this essentially a thermodynamic kinetic problem?
So heat does inhibit DNA repair. Hyperthermia is a very powerful DNA repair inhibitor. In fact, it was a big back in the 80s and 90s as a promising treatment in combination with radiation therapy, because if you can deliver the heat at the same time you can deliver the radiation, you can really sensitize the cells. The problem is that after the heat's been delivered, with time, those cells can adapt to that heat shock and produce heat shock proteins. And those heat shock proteins can then act as chaperones to prevent further damage from heat. They can help protect DNA by free radical scavenging and packing the DNA's topology and structure in different ways. So yeah, heat is a very tricky thing. So when I was talking about the heat in terms of adapting responses, I was talking about giving the heat ahead of time, allowing the whole process to kick in, allowing the cells or the animal to become resistant because it's produced all these proteins that protect it from subsequent radiation or heat. Now, I think just trying to remember the experiments we did, you can actually take cells that are deficient in DNA repair and give them heat shocks, and they will become resistant to heat, but not resistant to radiation. So it suggests that the heat is not inducing a DNA repair mechanism; it's protecting in a different mechanism. So take any cell that's deficient in a type of DNA repair and give it a heat shock, and then ask the question: do they become resistant to radiation? Have we induced repair? And some will become resistant, but not because of DNA repair induction, because we knocked out the genes for repair.
Thank you, Doug. So we have a question regarding comparing space and underground. So the question is as follows. How do the findings from SNOLAB's shielding environment and NASA's BioSentinel mission complement each other in understanding DNA repair mechanisms? And what implications do they have for both space radiation protection and improving cancer therapies on Earth?
That's a really good question, but it's a tough one too because it's about 5 questions in one. Um,
Yes.
I'm gonna try and answer that, as simply as I can, and that is, we don't understand. I think the most fascinating thing that I'm dealing with right now is that I've been in this business for a long time, and I've never really considered using an anhydrobiotic model to look at how cells respond to radiation damage, whether it's from space quality radiation or whether it's from no radiation underground or whether it's from radiation we use in the clinic, whether it's flash dose rates or stuff like that. And using this yeast model system is really telling us a lot about what the role that the initial DNA damage plays in how the outcome of the cell in terms of survival or transformation happens later. So I think what's going to happen, in at least my my expectation, at least what we're doing with this model system underground and with the BioSentinel stuff that they're doing at NASA, is that they're going to complement each other in a way that NASA's going to have dose rates and survival and things like these yeast will be sitting on the lunar surface, for example, collecting and accumulating the types of lesions and damage that you get from cosmic radiation without shielding from atmosphere. And we're working in our labs taking away that stuff or using other radiation sources to look at the mechanisms of how these cells deal with that. And once the data comes from sort of the yeast that may go back into space again, deep space, or on the lunar surface, or to Mars, they should complement each other with respect to how we'll be able to look at the mechanisms and the pathways that are turned on or off, and how that translates to what humans might experience in space and going to Mars. So, I mean, that's sort of the big overall picture. I don't know how we're going to complement NASA when we're putting yeast in the core of a nuclear reactor, but we're certainly surprised that they can withstand that level of neutrons and still survive. Some of them are surviving. So, and the dose rates are incredible too. So we've got a lot to learn in this model system, but it's a very powerful model system because it avoids a lot of the issues we have with growing cells that are alive and are replicating and need to be fed and need oxygen and CO2 and all that kind of stuff.
So related to that, Doug, and for those that are interested in space radiation biology, the crux of the issue is what is the confounding factors imposed by microgravity or changes in gravitational fields. So I think there were some experiments, even with centrifuge on board of satellites and Russian satellite bringing yeast on it. But maybe you can comment on the potential effect of different levels of gravity on this exposure, radiation exposures in yeast.
I think those experiments have been done. I don't know the answer yet to that. I know that on the Artemis 1 rocket, there was also an experiment being done with yeast that were going to actually come back after the mission, and then the this yeast cells were then going to be analyzed from a genomic perspective to look at what the effect of microgravity and radiation was, because they were on the rocket that went out there and they came back. I think it was 10 days or something. So I'm not sure where that data is right now, but and I don't know the literature. I should know whether they must have had desiccated yeast on the. I know they had desiccated yeast on the International Space Station in microgravity. I know that those yeast cells came back and were analyzed, but I don't know what the answer was. I don't know what the effect. I don't know if whether they discern between the radiation effects versus the microgravity effects. And as you know, microgravity is the biggest challenge we have when it comes to physiology and human health, right? Because it's really messes up everything.
Yep. More questions. Mary Helen Harper sent a question and said, excellent seminar, thank you. Can you speculate on the mechanism that might be at play with the high growth rate of cells with flash radiation of 55 grays per second?
Yeah, can I speculate on that? We've had discussions on this. Yeah, I don't know. I can't speculate on it. I mean, if someone else out there is listening and can type in what they think, I'm glad to listen to it. But we're trying to figure out why that would be. In fact, we were discussing this the other day, as if we could do this experiment possibly with yeast mutants that have defective metabolic pathways that are directly related to growth. We might be able to figure out if flash versus conventional radiation are differentially turning on genes that allow, enhanced doubling times or reduced doubling times. So we were just having this discussion the other day about how we go from this observation of why the things are growing faster to, to what the mechanism is. And I think we're going to be able to do that with various knockouts or even transcriptomics on which genes are getting turned off and on under these conditions. I don't think anyone's looked at why the growth rates would be different, even though survival is the same. Presumably, it has something to do with metabolism, maybe mitochondria, maybe defects or damage to mitochondria is less on flash dose rates, so you have more metabolism going on, better metabolism. I know that the growth rate's faster and the colonies are bigger, so something's going on there.
We have a somewhat related question. Concerning your work on the flash effects, are you contemplating experiments where normal or mutated yeast cells are exposed to very high dose rates of proton or neutrons at the variable oxygen tensions?
Yes.
That's a research project.
That's exactly what we're doing. Whoever said that question that's a great question, because that's exactly what we're trying to figure out. And we have a postdoc out at Triumph working with the various qualities of radiation and the dose rates. We're looking at presence and absence of oxygen and other types of environmental things that can change the types of lesions you get in DNA, absence, presence. There's a whole bunch of things we can do with this model system now that. I mean, I believe that we could probably give a whole seminar on that alone, on what we're doing, but we'll save that for the radiation research meeting, I think.
I think after your answer, we can say hi to Edward Hassan. He's listened to it.
Yeah. Hey, Ed.
Okay, we have more questions here. 1 second. Do you have a method of measuring ROS explicitly? Or is there a way to knock out natural ROS responses so they seem suicidal and measure only induced ROS?
Yeah, we do have those tools. I think our challenge right now is that we don't have enough people doing the research. If you know what I mean. We have collaborators, but we need more collaborators that can help us with these kind of things. It's the nice thing about the model system is that it's fast and you can reproduce it a lot. There's lots of reproducibility and it's very cost effective, not very labor intensive. Well, I say that, but you can maybe ask Mike or the other students to do it. It's pretty labor intensive counting the colonies and stuff. But yeah, no, I think all these types of questions are really important, and we do know that we can measure ROS directly with various endpoints. We just have not got around to do it yet. Need some more
Okay.
people. Need some more. If there's graduate students listening and they're interested in this, send us your names. We need some more graduate students and postdocs.
I think there is some people in the audience interested in that too. They are looking for there is opportunities for postdocs and graduate students to work on SNOLAB and those topics. Do you have
Yes.
some? Okay, so maybe that person will contact you. We will try to facilitate that. Okay.
Yep.
An
Yeah, they
interesting
can.
question.
They'll have my
I'm sorry.
email address. No problem.
Sure.
Just Google me at Nostrum and they'll get my email address.
Excellent. So another interesting common question. I will read it. Beyond there is an email before regarding the backlash that some scientists have when we're talking about hormesis and the beneficial effect of low dose of radiation. Beyond, do you find backlash in other radiation spaces by other radiologists, radiobiologists, for example, NASA investigators? How do you move forward or find funding and support for something that has been very controversial?
Yeah, this is well, I think I don't think you heard me say the word hormesis, right? I just called it adaptive response. And we're actually trying to figure out mechanism of what's going on with these low doses. And like I said with the stuff that Ed Azam did, where even a single photon was having the same effect. The, I don't tend to get a lot of pushback when you talk about hormesis. In fact, you're starting to see more and more organizations, like for example, the Health Physics Society. They're making different statements about where we should draw the line with respect to low doses and high doses, i.e., low dose risk versus high dose risk. And so more and more people are accepting that. I think a lot of the regulatory bodies are basically their hands are tied in the sense that they have to have something to regulate too. And right now, the model of the alarm models, low reasonably achievable, and the LNT model is argued to be a model that is useful for radiation protection. I would argue that that's not the case because it's costing us a lot of money to protect people from photons of radiation that are insignificant biologically. So I would suggest that setting a threshold for doses, tiny doses that below that don't matter, would be cost effective and benefit health and humans in a different way, save money, better investments this time. So yeah, and like I said, and there's people that even are scientifically knowledgeable that still have a belief that it's all bad, and there's nothing you can do about that. That's just the way it is, and that like that little quote I put at the end is just an example of it doesn't matter what people see in terms of the data. Some people just believe it, and if they believe it, it's basically you're not going to change their opinion. And sometimes we can change opinions, but there's whole articles written on why people are resistant to the idea that low doses of radiation might not be harmful, and there's a whole sort of hypothesis and theories of why that would be. But they believe that opinions are one thing, but when it comes to the ability to understand, or the opinion of that all radiation is bad and that no matter what you tell me is more of a behavioral thing. And that's a behavioral thing. If you look at some research that's been done, that it's sometimes easy to change opinions, almost impossible to change behavior. So that's where you just have to sort of say, well, we'll just keep doing the research, and as the tools we get better and better and better. Like I thought we had it all figured out when I was working at CNL back in the 80s and 90s. It was pretty simple. There's this thing called messenger RNA that made a protein, and that was it. DNA, RNA, protein, but now it's way more complicated, and the tools are getting more and more powerful. So we just need to keep people in this business, right? We got to keep recruiting young people in radiation sciences, especially with this whole thing of revive nuclear energy and space travel. We're going to need a lot more people getting into this business, and it comes down to that issue that someone brought up before about getting funding. I mean, yet you can't actually say you're going to do research on hormesis. You have to say something like you're going to look at the mechanisms of how photon interactions work at the level of DNA or something, and then whatever the answer is, the answer is. So you have to be a bit not creative in the way you present some of these grants.
And also a principle to be agnostic and saying that the data will tell you where are you on that. So There's a comment, there's a faculty professor saying that my graduate student and I were both shocked at your first graph of survival rates for rats above the LD 130 lows. The presentation was excellent, but he was very impressed and surprised by that data.
Well, that paper, and the thing is, that paper, if you just go on to Google search or PubMed and ask the question, what does heat do when it comes to radiation resistance? That thing doesn't come up, and it's hard to find papers that. In fact, the AI, if you go to ChatGPT, and you ask that question, can I heat a mouse up, and it'll go into a long explanation, but it won't find that paper. And I find that phenomenal that you can actually go from 100% lethal to 100% survival by having a fever the day before or taking a sauna the day before, or it's crazy. And when you think about what LD50 means, if we take everyone on this WebEx here and basically say, give them all the same dose at 4 gray, why are half of them going to die and why are half of them going to stay alive? It's probably because of a lot of environmental factors that we did yesterday or, a couple hours ago. They're going to change the way our whole body responds. And I agree with whoever said that when I first seen that paper, I went, this can't be true. But I mean, that's the data. That's the way it happened. It's a massive amount of radiation protection. And it would be nice to be able to look at what mechanisms were going on under that situation. The problem is LD50 or LD100 experiments right now with mice, that it really is not very ethical to be doing those experiments. And I think the ethical review boards would shun them and not probably give them approval. Although there are ways of not doing the LD50 itself. If we know they're all going to die in 15 days after the 9 gray, we have to figure out how we can maybe look at the mechanisms of that. But it's truly a phenomenal thing to see that kind of resistance.
It's an interesting comment here that say it's time to install a sauna on the International Space Station. Maybe that would be that's
Well,
nice comment.
it's funny because Ron Mitchell and I used to joke about that when we were doing these experiments, to be like, just turn the heat up. But the problem is if the solar flare is coming and you're turning the heat up at the same time, you're going to radio sensitize everything. So you got to kind of know ahead. And I don't know what the impact of repeated, say you said I'm going to have a sauna every day. I mean, these are really interesting experiments that some people need to do when it comes to radiation response. And the thing is, if the same thing happens when it comes to cancer risk, we should start looking at how, heat and hyperthermia might be turning on mechanisms that can reduce cancer risk and extend longevity.
Yeah. 1 second. I think we have a new post. Okay. Could I get your thoughts on increasing population epidemiology research coming out, for example, on nuclear workers, with evidence of a linear dose response relationship for solid cancers?
Yeah, like I said, I've seen those papers. They're coming out, and there'll be another paper that will challenge it likely and look at the confounding factors and look at all the other things. So I'm not going to comment on the whole epidemiology LNT model stuff because that's not what I work on.
So another question. Can you please comment on approaches to integrate radiobiology with epidemiology to reduce uncertainty of predicting risk at low dose radiation exposures? The $10 million question.
Yeah, that's something that people talk about and we have to do, right? I mean, I think one example I have, and I might have mentioned it briefly, but the whole idea of using epidemiology, say in uranium miners or gold miners, to look at the effects of radon and trying to tweeze out the confounding factors like in the non-smokers. What about the dust? What about the diesel fumes? But I don't know if anyone's accounted for the fact that many of these miners were literally forced to inhale nanoparticles of aluminum oxide, and not even nanoparticles, but dust right down to nanoparticles. And this so-called McIntyre powder was put into the air, into the change rooms, and the dries, and in the cages going down. So you're forced to breathe it. And it was good intentions because the idea was this aluminum will help expel the silica dust when you're mining, and that will reduce the risk of getting silicosis. But we know that silicosis itself causes inflammation, which is a tumor promoter. We know that aluminum oxide has a lot of effects when you combine it with other dust, and silica fibers, and then you throw in radon on top of that, and now you're trying to figure out, is it the radon that's causing this lung cancer, or is it all these other things that we never really accounted for? And I think that's, as we do the biology, and in fact, what we started to do was look at what is the transformation risk if you give radon and combine it with aluminum powder. We have to find out what that is, and that's one of the things we're going to be doing in our radon chamber, is looking at the combined effects of these things the miners were put through while they were mining. But it's tough. The biology, it's tough to say what's going to come first. The epidemiology will point the finger in the right direction, and then the biology has to follow and say what's going on here, like what mechanism would be to make this happen. So,
well, as I hear from great radiobiologists, biology is biology. So the complexity of the equation is amazing. So we are getting close to the end.
Yeah, I was gonna say I start charging extra after 120. So,
So let me ask you this. For example, suppose that in the radon effects arena and risk and all kind of potential risk factors and associated human risk, if you have unlimited possibilities in terms of funding, and you're looking for a breakthrough approach in how to deal with the radon, what areas do you think that will create a good breakthrough in terms to protect people from radon, or prevent the effect of the radon, or create some countermeasures if it's necessary to deal with the radon risk?
well, I think there's there's a lot of that's one of the most controversial ones that I can think of, is the whole the notion that radon's the second leading cause of lung cancer, or the number one cause of lung cancer in non-smokers. And so I think what we need to have is more biology, and that's why these radon, you're building one at CNL, right? Your radon chamber, there's going to be more radon work being done. So I think people have been sort of, I would say there's mitigation strategies for radon, right? You just get the radon out of your house, reduce it to a level where this doesn't really cause any problems. But I don't think anyone's shown that even at the higher levels of radon in the house, which is actually low levels, where the regulatory limits are set, that actually has a detrimental effect. I think it's still all extrapolation from high doses. And so until we get the real data, this shows us some real risk. I don't know. I think radon will still hang around, but I think the fact is we're building instruments that'll test that. And that's where the biology can help with the epidemiology to help figure out what are some of these confounding factors and stuff.
Okay, so we need to continue with the usual approach, or you see new breakthrough approaches to dig in on this basic data gathering?
I don't know. I can't, Marcel. It's a tough one with the radon one, because, as you know, there's lots of data out there that says it isn't harmful for you. It's just a matter of which data you look into, and you look at some of the work done by Bernie Cohen back in the 90s, where he looked at all these different counties across the United States that have high levels of radon, and then looked at the frequency of lung tumors per 10,000 people, and he got an inverse dose response at the low dose end. So, the higher the low doses of radon, the less lung cancer risk there was. So that data is out there too, and it just depends on which data people want to look at and which data they want to use to make their case, whether it's good or it's bad. That's been that's been that way all since the beginning of time when it comes to radiation. You just pick people pick their sides, and then pick the data. What's it called? The conformational bias or something, where you pick the data that matches what you think, and even if it's the same data that someone else has interpreted a different way, you get this conformational bias between people that do different types of work and stuff. So,
Well, thank you, Doug. I think it's the time to wrap up. today. And Doug, that was fantastic webinar. We appreciate very much your contribution. It was very exciting. I think a lot of people got inspired by your talk. And we are looking forward to continue the discussion. But we are very, very honored to have you today. And please, if you have more questions, you can forward to me, and I will try to forward to Doug, and we will continue the discussion. Doug, thank you
Yeah,
very, very, very much. It was
we
fantastic.
can we can continue the discussion in San Juan if people want to come and come to the radiation research meeting.
Exactly right. So, okay, thank you very much for all to attend this meeting, and we will continue in the near future with new talks, with new interesting ideas and data to continue the discussion of low dose radiation effects. So, thank you very much, and I think, Doug, if we can stay for a minute, and maybe we have a debriefing after that. But
Here.
thank you very, very, very much.